Hello guys, welcome to my channel. So as you know, we are studying about the cell cycle. That's our cell control system. We studied about different mechanisms that are controlling the cell cycle through G1 phase, S phase, right? And now we'll move to the last phase, that is the M phase, and we'll study mitosis. All right, so let me we'll leave these things, remove this here. Just all these things. All right. So we'll study now mitosis, right? That is with the M phase, and M phase also has cytokinesis as well. So mitosis and cytokinesis too. All right. So first, in this, as we are studying about the cell cycle, right? We studied G1 phase, S phase, right? The DNA is duplicated, replicated here. The chromatids that we studied here, right? The two sister chromatid forms, and they are held by the cohescence and condensance protein, right? But before the M phase actually begin, there are two critical events must be completed by the cell. DNA must be fully replicated before the M phase begin. DNA should be replicated, and in animal cells, the centrosome must be duplicated as well. So two events must be completed. That is, DNA must be replicated and centrosome should be duplicated. So what does this mean, centrosome should be duplicated? That means, so during, for example, we'll show you. For example, this is the nucleus, right? And it's in the G1 phase, right? For example, and here, for example, this is the centrosome in G1 phase. Right, if you don't know, centrosome is the principal microtubule organizing center in animal cells. So it's an organelle as well, centrosome, but the main function is it's a microtubule organizing center. So what the centrosome does before going to proceeding to the M phase, so the centrosome duplication starts at the same time as the DNA replication start, and this, like in the G1 and then the S phase, right? And this centrosome duplication is also allowed by the same the CDKs, where we have written like the SCDKs or G1 SCDKs, right? G1 S CDKs, right? Cycle independent kinases, right? So this is also initiated by, this allows the DNA to replicate, and this also allows centrosome to duplicate. So for example, this is G1 or stage, and we have something like this nucleus, and then again, so like for example in the S phase, or it will be something like S and G2, it would be something like this. So it start duplicating itself, right? This centrosome. And then in M phase, for example, like this is our nucleus, right? Sorry, so it will go like this, and it has now chromatids, right? Chromatids will be transferred, and so like first it's a single centrosome, then it start duplicating. Then, for example, it separates, right? It separates, it divides, and in this total M phase, entire M phase, actually, so then what it does, so as the M phase for the mitosis begin, however, there's this two centrosome will be separate, and each nucleates a radial array of microtubules. So this will two both will be separated, and these are the microtubules. They are arraying, and it's known as aster, right? And this the two ester, then this two ester, like what it will do, the two ester move to the opposite sides of the nucleus to form a two poles of mitotic spindle. So right, this has our sister chromatids and everything. And what will happen, so this will start, for example, I will just directly draw here, like this one goes here, the centrosome, and this, right? So it will be, so it will be something like this. So these two esters, this will start moving to the opposite side, right, of the nucleus, and to form the, and it will form two poles. It's kind of two poles of the mitotic spindle, right? So, and the mitotic spindle helps to separate the two sister chromatids, and this is how the each daughter cell will receive chromosome of itself, right? And the process of the centrosome duplication and separation is also known as the centrosome cycle. So it's just a simple kind of cycle, right? So centrosome is there about the nucleus, and it starts duplicating itself. Then the esters are formed like the microtubules, and then it started to move on the both opposite side of the nucleus, forming a poles of the mitotic spindle. Right, so this is the process how the centrosome is duplicated. So before the M phase begins, so this two things will be done. So that it should be duplicated. And the mitosis, so the mitosis itself is divided into 5 phases, right? So, well, I know this, we don't need this Down syndrome right now, right? So mitosis, oops, I think I'll do this. Mitosis has 5 stages, right? So number one, that is prophase. Number two is prometaphase. Phase. Number three is metaphase. Number four is anaphase, right? And number five is telophase. Telophase. This is how the mitosis is divided itself in the 5 stages, and after this telophase, cytokinesis will begin. Right. So first we'll study about the prophase. Prophase. Right. Right. So the first is the prophase, and what happens in the prophase? So the mitotic spindle begins to form in the prophase that we saw here. So this is happening in the prophase. So this assembly of highly dynamic spindle depend on the remarkable properties of the microtubules, right? So the microtubules continuously polymerize and depolymerize by the addition and loss of the diatubulin subunits, and the individual filaments alternate between growing and shrinking, a process called that is called a dynamic instability. But at the start of mitosis, so what happens, the stability of microtubules decreases. Why? Because of the mcdk, like this SCDK. Because of the mcdk, the stability of microtubules decreases, right? Because the mcdk phosphorylates microtubule associated proteins that influences the stability of the microtubule. So as a result, during prophase, rapidly growing and shrinking. So rapidly growing and shrinking as well, microtubule extend in all directions from the two centrosomes, exploring the interior of the cell. So some of the microtubules growing from one centrosome interact with the microtubules from the another centrosome. So for example, this is one centrosome and this two. So there are microtubules growing, right? So there are like sometime interaction as well. All right. So there are sometime interaction. Some microtubules growing from one centrosome interact with the microtubules from the another centrosome. So this interaction stabilizes the microtubules, preventing them from depolymerizing, and it joins the two sets. So these two sets will be joined of microtubules together to form a basic framework of mitotic spindle with its characteristic bipolar shape. So the two centrosomes that give rise to the microtubules are now called spindle pole. So this is now, it's called spindle pole from both sides, right? And the interacting microtubules, so this interacting microtubules are called interpolar microtubules. So they are called interpolar microtubules, right? So this is what this happens in the prophase of mitosis, right? So that was prophase. So in prophase, this mitotic spindle develops, right? And now the second, so that is prometaphase. Should I write it down here for the prophase, like what happens? Just a short line. Write down mitotic spindle. So in prophase, I will write it down. Mitotic spindle starts to assemble. So this happens in prophase. All right. So now prometaphase. So what happens in prometaphase is, so prometaphase starts abruptly with the disassembly of the nuclear envelope, so which breaks it up into small membrane vesicles. So I will show you. Let me remove this part. We don't need this. So for example, this is the cell that is going through, and inside the cell there is the nuclear envelope, and inside we have our chromosomes, right? So for example, it's going to the mitosis, so the prophase has begun, so it has this, right? So I am just drawing, not overlapping it. So this is our centrosome that has formed. So this happens in prophase, right? But in prometaphase, so what happens? So this is a nuclear envelope. I will write it down here. This is our nuclear envelope. So what happens in prometaphase? Prometaphase starts with the disassembly of the nuclear envelope, and in the prometaphase, this is actually a round, right? It covers the chromosomes. So what happens in this? It disassembly and it breaks up into a small membrane vesicles, right? So this process is triggered, and how it is, this process is done, how it is break into small vesicles is actually done by the phosphorylation and consequent disassembly of nuclear pore proteins. So the proteins and the intermediate filament proteins of the nuclear lamina, the network of fibrous protein that underlies and stabilizes this nuclear envelope. So for example, so this looks like this, and after that, when it breaks apart, so it actually, like, it may be just like this in the fragments, and now the chromatids that we have is actually like outside of the nuclear envelope, and that is available to grab by this centrosomes. So the spindle microtubules, right? The spindle microtubules which have been lying in wait outside, right? It was waiting outside. So when the chromosome, the nuclear envelope breaks and the chromatids are outside, they can grab the chromatids. So the spindle microtubules grab hold of the chromosomes, right? The chromosomes that we draw here at kinetochore. So, all right, so I will write it down here is a point. So the spindle microtubules grab, so this grab hold of chromosomes at kinetochore. So what is kinetochore? So this is a protein complexes that assembles on the centromere of each chromosome. So for example, you know the centromere, right? So this is the center part of this is our centromere, right, of chromosome. So this kinetochore, like this we have, so I will show you the two sister chromatids we have here, right? So this kinetochore is actually built at the center of like this, so we'll point as kinetochore, right? So this is kinetochore. Each duplicated chromosome has two kinetochores, right? Each has two kinetochores, one on each sister chromatid. So these are our two sister chromatids, both having each kinetochore, which faces in opposite direction. It should face in opposite direction. So kinetochores, what happens? The kinetochores actually this recognize the special DNA sequence in the centromere, and if the DNA sequence in the centromere is altered, kinetochore fails to assemble and consequently the chromosome fail to segregate properly during the mitosis. Right? So the most important thing is the DNA sequence should be properly so that the kinetochore can recognize and assemble itself on the center of the chromosome, right? So once this nuclear envelope has broken down, a randomly probing microtubules, so the randomly probing microtubules encountering, so this will be kinetochore, this will encounter the kinetochore and it will bind, thereby capturing the chromosome. So for example, I'll show you the bigger picture. So remove this here. For example, this is our we have some, these are our nuclear envelope that has been broken down, and for example I will just draw two. And for example we have this, right? Something like this is just a random picture, but this is how when the nuclear envelope is broken down the kinetochores are formed. So the kinetochores microtubules link the chromosome to the spindle pole. This is how it is. Because kinetochores of these two sister chromatids face in opposite direction, they tend to attach to the microtubules from opposite poles of the spindle, so that each duplicated chromosome become linked to both the spindle poles, and the attachment of the opposite poles, that's called bioorientation, generate tension on the kinetochores which are being pulled in opposite directions. So this tension signals to the sister kinetochores that they are attached correctly and are ready to be separated, right? So when they attach, so there will be a tension, and when there is a tension, so both of this, as they are both together, right, held with the cohesions, so when the tension arrives, so both will know, okay, so the tension is there and the tension is here as well, so they will know, okay, now we are perfectly attached with this end of the mitotic spindle, right? Right, so basically there are three types of microtubules. So the one that is outside, so that are called aster microtubule, right? And as we named here before, aster microtubules. The one that are joining or overlapping each other, right? Overlapping each other, they are called interpolar microtubules. And the one that is actually attaching to the kinetochore, that are called kinetochore microtubules. Right, so this thing happen in the pro-metaphase. Then after pro-metaphase, there is a metaphase. So metaphase is actually just aligning this two separated chromatids. So during the pro-metaphase, as we saw, the duplicated chromosome now attached to the mitotic spindle, right, begin to move about, right, as a jerk the first this way, and like it's like a tug of war between two centrosomes, right. Eventually they align at the equator, and after like pulling from one side and pulling from other side, eventually they will align at the equator of the spindle halfway between the two spindle poles, thereby forming a metaphase plate. So once they are in the exactly at the equator, it will form a metaphase plate. And this event defines the beginning of metaphase actually. So although the forces that acts like to pulling to bring the chromosomes to the equator are not completely understood, but both the continual growth and shrinkage of these microtubules and the action of microtubules motor proteins are required, and the continuous balanced addition and the loss of tubulin subunits is also required to maintain the metaphase spindle. Right, so the chromosome gathered at the equator of the metaphase spindle oscillates at back and forth, continually adjusting that position, indicating that in the tug of war, right? So it will continue to adjust until they are at the equator. If one pair of the kinetochore attachment is artificially severed with the laser beam during metaphase, the entire duplicated chromosomes immediately moves towards the pole to which it remains attached. Similarly, if the attachment between the sister chromatids is cut, the two chromosomes separate and move towards the opposite pole. So this experiment actually show that the duplicated chromosomes are not simply deposited at the metaphase plate. They are suspended and they are under tension. So these two are suspended under the tension of this microtubules. So in anaphase, the tension will pull the sister chromatids apart. Alright, so that was metaphase, and after metaphase there is the anaphase. So anaphase also begins abruptly with the breakage of this cohesion linkage that hold the two sister chromatids together. Right, so now the step begins where the holding protein that is holding the two chromatids that need should be break. So this releases each chromatids together. Sorry, this release allows each chromatid now considered a chromosome. So they are before chromatids, now they are considered chromosomes when they are separate and to be pulled to the spindle pole to which it is attached. So this movement segregate the two identical sets of chromosome to the opposite ends of the spindle pole, right? So the cohesion linkage is destroyed by a protease called separase. So the cohesion, this linkage is destroyed by protease enzyme that is called separase. So this enzyme becomes active in anaphase. So before anaphase begins, this protease is held in an inactive state by an inhibitory protein that is called securin. So for example, if this is our what we call, this is our separase, right? So this is held by a protein. This is held by a protein that is called securin. And this is inhibitory protein. So this is held by a protein, and this is called inactive separase because it's held by this protein that is called securin. So at the beginning of anaphase, this securin is targeted for the destruction by APC, anaphase promoting complexes, right? The same protein complex, if you remember, we discussed that marks M cyclin for degradation as well. So this protein anaphase promoting complexes target this securin, and once the securin is removed, the separase will become active and free to severe the coherence linkage, right? So once the sister chromatids separates, the resulting chromosomes are pulled to the spindle pole to which they are attached, and they all move at the same speed, which is typically about 1 micrometer per minute. The movement is consequence of two independent processes that depend on different part of the mitotic spindle. So there are like two processes called anaphase A and also anaphase B. So it has anaphase A and anaphase B, right? And this both occur simultaneously. So like in anaphase A, the kinetochore microtubules shorten, right? So the kinetochore microtubules, they shorten, right? And the attached chromosomes move forward. So this attached chromosome will move. This will move on this side and that will move on this side, right? In the anaphase B, the spindle poles themselves move apart. So first, in anaphase B, this becomes shorted, right? And so this will go on this way. So for example, this become shortened. So I will show you understand that, right? This becomes shortened in anaphase A. So actually this is moving this way. This becomes shorter and it will try to pull. And in anaphase B, this actually the spindle pole themselves move apart. So this actually goes on this side and this on this side in anaphase B, and this is in anaphase A, right? And separating the two chromosomes. So the driving force for the movements of anaphase A is thought to be provided mainly by the loss of tubulin subunits from both ends of the kinetochore microtubules, and the driving force in anaphase B are thought to be provided by two sets of motor proteins, the members of kinesin and dynesin families. So this motor proteins provided for the anaphase B, and the kinesin proteins act on the long overlapping interpolar microtubules, right? Interpolar microtubules means that they are joining together from the opposite poles, right? Past one another equator and pushing the spindle poles apart, and this dynein protein anchored to the cell cortex that underlines the plasma membranes pull the poles apart. So these are the two processes that happens in anaphase, right? And one more thing, so an unattached chromosome will prevent sister chromatid separations. What does that mean? So if a dividing cell were to begin to segregate its chromosome before all the chromosomes were properly attached to the spindle, if one daughter cell would receive an incomplete set of chromosomes while other will receive the surplus if they are not attached properly. And in either of the cases it would be really dangerous. So thus a dividing cell must ensure every last chromosome is attached properly to the spindle before it completes mitosis. So to monitor chromosome attachment, the cells make a use of a negative signal. Unattached chromosomes send a stop signal to the cell cycle control system. So if the chromosomes are not attached properly, it will send a negative signal to the cell cycle control system. Although only some of the details are known, it's not very known mechanism, but the signal, what we know is the signal inhibits further progress through mitosis by blocking the activation of APC, that is the anaphase promoting complex, and without the APC the sister chromatid remain glued together. Why? Because the APC, that is the anaphase promoting complex that target the securin and inactive separates become active, acting on this chromosome, and if this is not present it will not happen, and this is thus none of the duplicated chromosomes can be pulled apart until every chromosome has been positioned correctly on the mitotic spindle, and this is so called the spindle assembly checkpoint. thereby controls the onset of anaphase as well as exit from mitosis. Right, and the last phase is our nuclear envelopes forms, that is in our telophase. So by the end of the telophase, right, by the end, the two daughter of the chromosomes, the daughter chromosomes have separated into two equal groups, one at each pole of the spindle, and during telophase, the final stage of mitosis, the mitotic spindle disassembles. So this spindle disassembles in telophase and the nuclear envelope that is broken down also reassembles around each group of the chromosomes to form the two daughter nuclei, right? So the vesicles of the nuclear membrane first cluster around individual chromosomes and then fuse to reform the nuclear envelope. And during this process, the nuclear pore protein and the nuclear lamins that were phosphorylated during the prometaphase are now dephosphorylated, which allows them to reassemble and the nuclear envelope and the lamina to reform. And once the nuclear envelope has reformed, the pore pump in nuclear proteins, the nucleus expand and the condensed chromosome decondense into the interphase state. Right, as a consequence of this decondensation, gene transcription is able to resume. A new nucleus has been created and the mitosis is complete, and all that remains is for the cell to complete its division into two separate daughter cells. Okay guys, so that was the end of the telophase, and after telophase, cytokinesis begin. And I think we'll do one more part. Okay, so I think this was enough for mitosis. I don't want to make a very long video, right? But we'll see in the last part of this video and we'll discuss cytokinesis, and that's all finishes our cell cycle and the cell cycle control system. All right, thank you guys for watching, and don't forget to subscribe